This is looking good. Make the changes and we'll submit the paper next week.
Great, I'll get it done.
Oh, and Pam, send in Dave. I like Pam. She's smart, productive. She'll be successful regardless of what I can offer. I see her as a future ally within the research community, so it's important that I be a good mentor for her now. But those other grad students, they're a different breed. Take a seat. What do you have for me?
I ran those gels you asked for, and to monitor the protein degradation for at varying temperatures, and
Dave is a hard worker, but he's no Pam. Grad students like him are here to produce results, publish papers, and get out. Some of them have dreams of running their own labs, curing cancer, winning a Nobel Prize. But that won't happen. And frankly, I don't care where they end up. I admire their tenacity, the desire to learn from me, to be me, to suck all they can from me. But sooner or later, you have to cut the supply. Can't feed them forever or they'll run you dry.
so what do you think I should do next?
Pathetic, naive creatures. They can barely manage their own thesis projects. And they're totally oblivious to the real reason they're here. You're all just data generating cheap labor. Welcome to grad school.
Weird. Oh shit. Not today. Undergrads. What?
Ha! I thought you said you were gonna get here early today.
Yeah, it was a long line at the bus stop.
Undergrads?
Yep.
Yeah, well, it's the first day of class. They're everywhere.
Yeah.
I know what you mean. All summer long, it's been peace and quiet. You can get a seat anywhere on the bus. Literally only took 4 minutes to get coffee. September rolls in, and bam! Undergrads everywhere. Invading your personal space and... What? I don't mean you guys. You guys are cool. For the record, no, they're not. Anita, let's go make some buffers.
So how's the paper going?
I can't wait till it's done. I feel like I've been sitting in front of this computer for months.
Yeah.
So, Ben's growing cell culture today.
Mm-hmm.
Oh, he's so nervous his hands are shaking. Didn't you tell him that the strain of E. coli we work with isn't dangerous? That would help calm his nerves.
Yeah, I did tell him. Great. Now it looks like he's afraid to touch it, like it's hot or something. Wait, did we autoclave fresh media this morning?
Yeah, like 6 liters or so. Why? Oh my god. Is he using hot media? He's adding culture to hot media?
And he just killed his culture.
We need to say something.
No, wait. He'll figure it out at the end of the day when nothing grows.
Seriously?
Well, yeah. If you tell him now, he won't learn. He'll learn when it doesn't grow.
Well, the flask must be hot. He better not drop it.
Alex, Has Janet replied to any of your emails recently? I sent her data a while ago and she hasn't replied about anything.
Oh man, he actually dropped it.
You fool! You got culture over my shoes!
I'm sorry. Get the bleach.
Wanna get some coffee?
Yeah, let's sneak out of here.
Hey, look, all I'm saying is that Jess is a good friend of mine, so please don't screw this up.
Okay, it's not like I intentionally screw these things up. Sometimes it just doesn't work out.
What the hell was that?
Oh, that was my phone. Reminding me of my comps.
All right, when's that again?
3 weeks from now my proposal's due, and then my presentation's at the end of October.
Guys, you don't want to go in the lab right now. It still smells like bleach. So Dave, what's this presentation you're talking about?
My comps.
Comps? Am I supposed to know what that is?
Yeah, comps, comprehensive exams, you know, the exam we all take to become PhD students.
Oh, comps. That's a cute name. You know, back in Britain we called them preliminary exams. I guess we just took it a little more seriously than you guys.
You know, just because you did your PhD in London doesn't make you British.
It makes me more British than you.
So, how was your preliminary exam?
Well, I actually didn't have to do it. My research was just so good. The university practically begged me to join them.
I don't believe that.
Excuse me, I'll be right back.
Oh, it's true. I even got a full scholarship.
How's it going, Ben? You setting up a cell culture?
Yeah, and about earlier, I'm sorry I spilled the last flask.
Oh, don't worry about it. The important thing is you weren't hurt. Good. It's not hot. So, the lesson from today that we can take is that we don't use hot media. And do you know why that is?
Because then the flask will be hot, and you're more likely going to drop it.
Yeah. Yeah, but there's a bigger, more important reason why we don't use it. Do you know what that is? Okay. What happens when you add cell culture to really hot media? What happens to those cells?
Look, Alex, there's no need to be jealous. We all work in the same lab, and as your colleague, I'd be happy to give you pointers on how to be a good researcher.
No thanks. I'm good for right now. Everything cool with Ben?
Yeah, I think it's just going to take him a while to get used to the lab.
My students settled in just fine. Maybe the problem isn't with the student, maybe it's the mentor.
So anyway, Janet finally got back to me. And she wants me to redo the whole assay for tomorrow. Oh, today's going to be a long day.
Well, don't forget your meeting with Jess tonight.
Yeah, about that. Do you think we can reschedule?
No.
So, I'm sorry, you were saying you don't know how many years of school you have left?
No, because it depends on how the research goes, really.
Oh. Well, how long do you expect that'll take?
Sorry. You wouldn't get that. That's a hard question to answer because it's not really up to me. You see, there's this committee of professors, and they decide when you've done enough work. And if they're satisfied with the work you've done, they will let you graduate. And if they hate your guts, then they will make you suffer longer.
Right. Okay. So, Alex tells me you study brain cancer. That's exciting.
Yeah, sorta. I study a few of the proteins that might be involved in brain cancer. Proteins are biological molecules found in the body. and they have specific functions.
I know what proteins are. I might not have a science degree, but it doesn't mean I'm dumb.
Sorry.
So,
Sorry.
what specifically are you looking to do with these proteins?
Well, it all started when I first read this paper that talked about brain cancer. And, then I ran an alignment and saw that the motifs were conserved throughout the homologs, and I was ecstatic. I mean, there was so much I could do with that. And that's when I ran into some problems, because I saw that mutations were not giving me the results I wanted. So I tried something different. So pretty much, I'd say 5-10 years down the road, when there's enough data and a concrete model around this, similar methods could be applied to a mammalian system, at which point it could be tested as a potential drug target for treating brain cancer. Well, after some, animal model experiments and human trials. Wow, I forgot how interesting my project is. Yeah.
Sorry, I zoned out about half an hour ago.
Too detailed?
A little.
Sorry.
Here's your receipt. Folks, have a good evening.
Thank
Thanks. Anyway,
Yeah.
okay, let's go.
Thank you.
Oh, I gotta go.
Now it's almost 9 o'clock.
Yeah, I'm sorry. I've got stuff at the lab I gotta do, but it was really nice meeting you.
Yeah, you too.
Okay, sorry I talked the whole time.
That's okay.
Okay, well,
Um,
sorry.
bye.
Yeah, bye.